Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wonder if we can tell if someone is going to develop a serious mental health condition, like psychosis, long before it happens? Well, this new study might have something to say about it. Today we're diving into a super interesting review about using brainwaves to predict who might be at risk for developing psychosis. Think of it like a detective story for your brain. Scientists are trying to find clues early on so they can help people sooner. So, what is psychosis? It is when someone starts to lose touch with reality, experiencing things like hallucinations or delusions. It can be really tough, not just for the person experiencing it, but for their families too. And the longer someone waits to get help, the harder it can be to recover. That is why it is so important to identify people who might be at risk before they experience a full psychotic episode. Scientists have identified a group of people called being at clinical high risk, or CHR for psychosis. This means they're showing some early signs like milder versions of psychotic symptoms or have a strong family history combined with problems in their daily life. It is like a warning sign. But here is the tricky part: not everyone in this high risk group actually goes on to develop psychosis. Some people might improve or stay stable. So the challenge is figuring out who really needs the most help. This is where our brainwave tool electroencephalography, or EEG, comes in. Basically, EEG measures the electrical activity in your brain. It is like listening to the chatter of your brain cells. Scientists can look at different patterns in these brainwaves to see if they're different in people who go on to develop psychosis compared to those who don't. They can look at brain activity while someone is just resting, or while they're doing a specific task like listening to sounds. One really common way to use EEG is by looking at something called event-related potentials, or ERPs. Imagine you show someone a picture or play a sound. An ERP is the brain's quick electrical response to that specific event. It happens within milliseconds. Think of it like a tiny electrical ripple in your brain caused by something you see or hear. Two specific ERPs that show up a lot in this research are Mismatch Negativity, or MMN. And the P300 MN is pretty cool. It happens when your brain detects something unexpected or different in a sequence of sounds. Even if you are not paying close attention, your brain notices the odd one out. It's thought to reflect how automatically your brain processes sensory information. The P300 is another ERP that happens a bit later than MMN. It's more linked to paying attention and recognizing something as important or a target. So MN is like your brain saying, "Ho, something's different," and P300 is more like, "Okay, I noticed that difference and it is important." Some researchers try to understand these brainwave patterns using a theory called predictive coding. The idea is that our brain constantly tries to predict what is going to happen next based on past experiences and what it already knows. When what actually happens doesn't match our prediction, our brain creates a prediction error. This helps us learn and update our expectations. In psychosis, it is thought that these prediction systems might be a bit out of whack, either overemphasizing predictions or underemphasizing incoming information. And MM and P300 could be ways to measure these prediction errors. So this study we're talking about today is a systematic review. That means the researchers went through a ton of other studies to see what everyone else has found about EEG and predicting psychosis. They wanted to see which EEG signals are the most consistent predictors. The researchers searched for studies that followed people who were considered clinically high risk for psychosis. They looked at their EEG data from the beginning and then tracked whether those people later developed full-blown psychosis or not. They needed studies that actually compared the EEG results between those who transitioned to psychosis and those who didn't. They included a lot of data and used statistical tools to combine the findings from different studies. After sifting through over 1,000 studies, they ended up focusing on 34 that met their criteria. What did they find? It wasn't a simple yes or no answer, as is often the case in science. The findings were mixed, but something stood out. The big winners for predicting who would transition to psychosis seemed to be MN and P300. They found that people who later developed psychosis often had differences in their MM and P300 signals. For these brainwaves, there were often medium to large effect sizes. Now, what is an effect size? Think of it as a measure of how strong the difference is between the two groups, the ones who transitioned and the ones who didn't. A larger effect size means the difference is more pronounced and likely to be meaningful. So, changes in MN and P300 were the most consistent signals across the studies. Looking at the details for MN, especially the duration MN, meaning the brain's response to a sound that is different in length, it was often reduced in the group that transitioned. For P300, specifically the P3B component, which is more related to attention, there were also differences, often showing reduced signals in those who transitioned. When they crunched all the numbers together for the ERPs, they found significant differences for several types of MN and P300. For instance, MN duration had a pooled effect size of 0.54, which is considered a medium to large effect. P3B, a part of the P300, had an effect size of minus 0.52, also indicating a significant difference. So, what does this all mean? The researchers are saying that while EEG is a feasible tool for studying people at high risk for psychosis, and many people tolerate it well, it is not a perfect crystal ball. One of the biggest challenges they highlighted is how diverse the group of people at clinical high risk actually is. People enter this stage with different symptoms, different backgrounds, and different life experiences. This makes it hard to find one single EEG signal that perfectly predicts who will transition. Another major issue is the methodological heterogeneity. This is a fancy way of saying that different studies did things a little differently. They might have used different ways to define clinical high risk or different criteria for deciding if someone had transitioned to psychosis. They also used various EEG tasks and ways of analyzing the data. This inconsistency makes it tough to compare results directly and build a clear picture. The researchers really stressed the need for data harmonization. This means getting everyone to agree on how to collect and analyze the data so that studies can be compared more easily. Imagine trying to compare apples and oranges if everyone called them something different and measured them in a different way. That is a bit like the problem here. They point out that there are many different questionnaires and tools used to diagnose someone as being at clinical high risk, and also to determine if they've transitioned to psychosis. This variation is a big hurdle. They also found inconsistencies in how studies reported things like whether participants were taking medication, which can affect EEG results, and if people had other mental health conditions along with their psychosis risk. This missing information makes it harder to draw firm conclusions. And something else they noticed is that most studies focused on simpler tasks, like listening to a sequence of sounds. They didn't often look at more complex cognitive functions, like memory or decision-making, using EEG. These more complex functions are also known to be impaired in psychosis, so focusing only on sensory processing might mean they're missing other important clues. To address these challenges, the authors propose a new framework called TRIDENT. It stands for Transdiagnostic Risk Identification using EEG with diagnostic tracking. The idea behind TRIDENT is to broaden the definition of who is considered at risk beyond just positive psychotic symptoms to include things like anxiety, depression, and declining social functioning. They also want EEG to be used as a tool to identify general risk for various psychiatric problems, not just psychosis specifically. TRIDENT also advocates for including assessments for common co-occurring conditions like anxiety and substance use, using more advanced EEG tasks that look at higher-level thinking. And of course, standardizing how data is reported so everyone is speaking the same scientific language. This would involve using common guidelines for data collection, analysis, and reporting of results. So to wrap things up, this review shows us that EEG has potential as a predictor for psychosis, especially signals like MN and P300. But we're not quite there yet. The path forward requires more consistent research practices, better data sharing, and a more unified way of looking at who is at risk and how we measure their brain activity. The TRIDENT framework is a great step in the right direction, pushing for a more standardized and comprehensive approach. It's about making sure that the valuable insights we get from EEG research can be built upon consistently, leading to better understanding and earlier help for those who need it. What are your thoughts on using brainwaves to predict mental health risks? Let me know in the comments below. If you found this breakdown helpful, give this video a thumbs up, and don't forget to subscribe to RUBIN'S Mind Lab for more fascinating explorations into the world of psychology and neuroscience. Thanks for watching, and I'll see you in the next one.